Hello there, I'm Adrian and today I'll be presenting about the current research and practices in cancer treatments. Many cancers can be cured and treated effectively. Chemotherapy remains a significant therapeutic option for a variety of malignancies, notably for primary, advanced and metastatic cancers, despite improvements made in surgery and radiation therapies. However, the cancer cells' inherent and acquired resistance to anti-cancer medications significantly limits the effectiveness of chemotherapy. The practical application of novel therapies has been made possible by novel delivery technologies, which have also greatly decreased side effects and allowed for the development of new and improved chemotherapy regimens employing currently available pharmaceuticals. Extracellular vesicles (EVs), which are crucial for the advancement of metastatic disease and the development of cancer, have recently attracted a lot of attention as effective medication delivery systems. On the basis of their biogenesis, EVs are divided into two groups. In particular, shared microvesicles (SMVs) with a typical size of 50 to 1300 nanometers are present in almost any extracellular body fluid and are responsible for the exchange of molecular materials between cells. Exosomes are small vesicles of around 30 to 150 nanometers that are derived from endosomes in physiological and pathological conditions and released by fusion of multivesicular bodies (MVBs) to the cell membrane. Exosomes have a role in the growth and spread of cancer, in the bidirectional connection between tumor cells and surrounding tissues, and also in the creation of the milieu required for pre-metastatic niche establishment and metastatic progression. Therefore, circulating vesicles are significant for clinical purposes in the diagnosis, prognosis, and follow-up of cancer. Exosomes can be extracted and used as anti-cancer vaccinations or nanosized drug carriers in cancer therapy, while they are also recognized as reliable diagnostic tools. Today, the early detection of biomarkers via non-invasive approaches is one of the major problems in cancer diagnosis. The monitoring of cancer growth and the effectiveness of therapy regimens should be possible with the collection of a sizable quantity of data both before and during tumor treatment. As markers for individualized medication, liquid biopsies have been utilized to find circulating tumor cells, RNAs, DNA, and exosomes. Thanks to the discovery of their contents, including double-stranded DNA, messenger RNA, microRNA, long non-coding RNA proteins, and lipids, exosome detection has recently been validated as a trustworthy tool for preclinical practice in various cancer types. Exosomes from several cancer cell types have been shown to contain double-stranded DNAs, and mutations in tumor-promoting genes, including mutant KRAS and TP53, have been found to be disease indicators. Exosomal AR-V7 mRNA has also been employed as a predictor of hormone treatment-resistant individuals with metastatic prostate cancer. A useful diagnostic technique is the gene expression analysis of many RNAs from urine exosomes. Colorectal cancer patients have been used long non-coding RNAs obtained from serum exosomes for disease prediction, and numerous microRNAs allow one to discriminate between several lung cancer subtypes. Exosomes that are GPC1 positive have been used to identify pancreatic cancer, and circulating exosomal macrophage migration inhibitory factor (MIF) has been used to forecast the development of liver metastasis. The presence of many lipids in urea exosomes has also been recognized as a prostate cancer indication. Furthermore, exosomes may also be used as biocompatible, low immunogenic natural nanocarriers for medication administration in the treatment of cancer. They can be actively loaded using laboratory procedures like electroporation and sonication, or passively loaded by combining pure vesicles with tiny medicines. The isolation of exosomes expressing transferrin receptor from mice blood has been investigated using superparamagnetic nanoparticles coupled to transferrin. They have been utilized to target liver cancer cells after doxorubicin incubation, reducing cell growth both in vivo and in vitro. In order to target the sigma receptor, which is overexpressed in lung cancer cells, a researcher modified mouse macrophage-derived exosomes with aminoethyl anisamide PEG. These cells were then passively loaded with a drug paclitaxel. These programs serve as targeting agents that could stop the spread of metastatic disease in vivo. There are currently 3 clinical trials using loaded exosomes for the treatment of various tumors. Phase 1 trial is assessing the ability of exosomes to deliver curcumin to normal and colon cancer tissues. Next, Phase 2 trial is examining the in vivo performance of autologous tumor cell-derived macrophages carrying methotrexate in lung cancer patients. Lastly, a clinical investigation is conducted to concentrate on autologous erythrocyte. Ad hoc exosome production techniques have recently undergone major developments. Exosomes are released by cells that have undergone genetic engineering to release exosomes with certain targeting molecules. Cancer vaccines have already been developed using exosomes from several cancer cells. In a Phase 2 clinical study for the activation of CD8 positive T cells in non-small cell lung cancer patients, autologous dendritic cell-derived exosomes with increased immunostimulatory activity were examined, with the disease stabilizing and a higher overall survival. Patients with colorectal cancer have received X-size-derived exosomes combined with granulocyte macrophage colony-stimulating factor (GM-CSF) in a Phase 1 study to induce a tumor-specific immune response. The defining of preclinical methodologies for isolation, quantification, storage, and common protocols for drug loading are mainly responsible for the many unresolved questions about exosomes clinical translation that still need to be resolved. To characterize their post-isolation half-life and to carry out conventional content tests, it's increasingly important to distinguish between cancer and healthy blood cell-derived vesicles. Innovative methods and tools have been developed for their aims, including microarrays, particular monoclonal antibodies, and RNA marker amplification techniques. Hence, in conclusion, recent advancements in cancer medical research have led to less intrusive, more accurate, and more successful cancer therapies. There are some distinct advantages and disadvantages of the main innovative cancer therapy approaches. Extracellular vesicle (EVs) is physiological secreted and good molecular characterization, high biocompatibility, and in vitro modifiable or loadable, while the application as cancer treatment is still questionable as it is still lacking of preclinical processes for isolation, quantification, storage, and drug loading. Thank you.